Today we're going to be talking about the cell cycle and how a cell is able to go through the process of mitosis. The cell cycle consists of two phases. The first phase is interphase, which is where the cell spends most of its time preparing for the second phase, known as the M phase. Interphase consists of three different parts. The first part is called G1, otherwise known as gap 1. This is where the cell grows in preparation for the next part. The following part is known as S, which stands for synthesis. The title fits its purpose as the cell goes through DNA synthesis, which is a fancy way of saying that the cell's DNA copies itself. G2, or gap 2, is the last part of interphase, which is where the cell grows even more and just enough to prepare for the next phase, M phase. Once the cell is done with interphase, it will move on to the M phase. And this is where an amazing thing called mitosis happens. Mitosis is the process in which the cell divides into two genetically identical daughter cells. By dividing, more cells are produced, which can help objects repair and grow. Mitosis can be split into another 4 phases. This may seem like a lot to take in, but one good way to remember is PMAT. P stands for prophase, the M stands for metaphase, the A stands for anaphase, and the T stands for telophase. To start us off, this is what the cell looked like as it was going through its functions during interphase. We have the centrioles right up here, and the material, which is composed of the cell's DNA, or chromatin, right here, surrounded by the nucleus. We are then led to our first part of mitosis, prophase. As we can see, the nuclear membrane is still visible, but it will start to dissolve in this phase, and the chromatin has turned into chromosomes, which are thickening and becoming more visible. The centrioles have changed a bit as well, and are starting to play a role in helping the cell divide, and we can see that between the centrioles, a spindle fiber bridge has formed. This will come in handy in the following steps. A good way to remember the prophase is the word prepare, as the chromosomes and the cell overall are preparing to divide. The following phase is metaphase. In this phase, we can see that the chromatids are now in the middle of the cell, and they were moved there by the now outreaching spindle fibers, which are released by the centrioles, which are now on the opposite sides of the cell. You can also see that the nuclear membrane has fully dissolved. An easy way to remember metaphase is by the word middle, as the chromosomes have lined up in the middle of the cell. Before we move on, I will also like to take into account that outer ring that surrounds the cell. That is the cell membrane, and keep a close eye on it as we move forward. In the next phase, known as anaphase, the chromosomes have been split, and we now have sister chromatids, which, pardon me, are supposed to look like this, and I modeled them wrong. Anyway, the sister chromatids are being pulled apart by the spindle fibers coming from the centrioles, and the cell membrane is undergoing preparation for upcoming changes. And a good way to remember anaphase is by the word apart. We are now moving on to the last part of mitosis, known as telophase. As we can see, 2 new nuclei have formed, demonstrating the 2 separate nuclei that the daughter cells will have, and the chromatids have uncoiled and are now back to their original state, chromatin. The cell membrane is demonstrating a cleavage furrow, which is the slight inward form that it is taking to eventually become 2 separate cell membranes. But wait! We're not done just yet. We still have to get through cytokinesis, which is the actual final step to this whole cell division thing. The cell membrane and the cytoplasm, which is the gel substance within the cell that surrounds the cell's organelles, has split, and there are now 2 separate cell membranes, 2 separate nuclei surrounding 2 separate but identical chromatin or genetic information. All this means that we now have 2 identical daughter cells. Thanks for watching!